Okay. So in this class we are discussing about these three topics. Okay. In the previous class we have studied about the fertilization process, right? So what are the events we have seen in the fertilization process? What are the different events we have studied in the fertilization process? The very first topic was to all the new students, this is your embryology class, okay. So in embryology, we mainly have two divisions. The first one is general embryology and the second one is systemic embryology. Mostly in the general embryology, we are going to study about the formation of 3 germ layers, and in the systemic embryology, we are going to discuss about the formation of different organs and other important structures, okay. So we are still in the general embryology topic. In the previous 2 sessions, we have discussed about the gametogenesis. Okay. We have spermatogenesis and oogenesis. Through the process of spermatogenesis, we are going to get the mature sperm or male gamete, and through the oogenesis, we will get the fertilized ovum or the female gamete. Okay. And in the second session, we have discussed about the fertilization process. Okay. In the fertilization process, we have seen the fusion of male and female pronucleus. So consider this as a female pronucleus and this as a male pronucleus. Okay. So after the fertilization, it is going to give us the zygote, which has diploid number of chromosomes. Okay. This is zygote. Okay. From this session, we are going to see what happens after the formation of the zygote. Okay. We all know after the formation of zygote, the cell will start undergoing the mitotic division. Okay. So this cell, the zygote cell, will start undergoing the mitotic division. Okay. Once the mitotic division takes place, we can see the formation of 2 cells. Okay. This is called 2 cell stage. 2 cell stage. Okay. Again, this 2 cell stage undergo one more mitotic division to give us 4 cell stage, followed by 8 cell, followed by the 16 cell stage, that is called as the morula. Okay. So now we have 4 cell stage, then followed by 8 cell, then 16 cell stage. Okay. So consider this as the 16 cell stage structure. Okay. So this is your 16 cell stage structure, which is called as the morula. Why the name morula? Because this structure somewhat looks similar to the mulberry fruit, so they named it as the morula structure. Okay. And the cells present at the morula stage are named as blastomeres. These are named as blastomeres. Okay. So what happens after the 16 cell stage? What happens after the 16 cell stage? Formation of implantation. Okay, fine. Next, is there any other cell divisions? This is 16 cell stage, morula. Yes, it will divide into trophoblast, embryoblast and other structure. Okay. So the 16 cell stage will give us this structure. Okay. something like this, then followed by okay. So now this structure is called as the outer cell mass. This is called as outer cell mass. Okay, and this part is called as the inner cell mass. Inner cell mass. Okay, the pole or at the point where the inner cell mass is present, I can say like this pole. This pole. Okay, this is called as embryonic pole. Embryonic pole. And the cavity present here, this cavity is called as the blastocele or blastocyst cavity. This is called as blastocele. Okay, any other structures here? So the division of your zygote takes place right after the fertilization. Okay, so this 2 cell stage to the 16 cell stage process is called as the cleavage. Okay, then from the 16 cell stage, this structure what you are seeing, this is called as blastocyst structure. Blastocyst. Okay, so this process is called as blastulation process. Blastulation process. So after the cleavage, we can see blastulation. Okay, so now the question is how the 16 cell stage directly turned into the blastocyst structure. Fluid. Okay, which fluid? Uterine fluid. Good. Next. Any other answers? How the 16 cell stage turned into the blastocyst structure? Remember, the fertilization takes place in the ampulla region, right? So if you remember the female reproductive system, this is the uterine tube or your fallopian tube. It has 4 parts. This is fimbriae and this is infundibulum followed by ampulla, then isthmus. Okay, mostly the fertilization, that means the fusion of your male and female gamete, takes place in the ampulla region. Okay, so when this cell division is going on, that means the cleavage process is going on, the fertilized cell, that is your zygote, move from ampulla to the uterine part or into the uterus or uterine cavity. This is called uterine cavity. Okay, so by the time it reaches to your uterine cavity, the cell will be actually in the 32 cell stage, not in the 16 cell, it is in the 32 cell stage. Okay, so the division does not stop at the 16 cell stage or at the morula stage. Remember this point. When the zygote reaches to your uterine cavity, it will be in the 32 cell stage. Okay, after reaching the uterine cavity, your 32 cell stage will undergo one more mitotic division to give us the 64 cell stage. Okay, later once the, now imagine this is the uterine cavity. Okay, now we have a cell which is in the 64 cell stage. Okay, all the cells are present exactly like this, even in the 64 cell stage. As soon as it turns into the 64 cell stage, the uterine fluid will start flowing into the cell. Okay, the uterine fluid will start flowing inside into this 64 staged cell. As a result, the cells present here will start showing some gaps like this. Okay, so these gaps are filled now by the uterine fluid. Okay, as the accumulation of uterine fluid increases, it will start pushing the cell towards the outer region. Okay, so whatever the cell move towards the outer region will be arranged as the inner cell mass. Okay, and all the fluid will go and accumulate in this cavity. Okay, the remaining cells will come and forms the bunch of group of cell as the inner cell mass. Okay, this is how exactly the 16th cell stage turn into your blastocyst part. So the 16th cell stage or the morula will undergo two more mitotic division, that is 32 cell stage and 64 cell stage. As it reaches to your 64 cell stage, the uterine fluid will start entering inside the cell. Okay, as a result, it will start moving the cells and the fluid will accumulate in one part. It will form the blastocele cavity part, and the other outer layer is called the outer cell mass, which is also known as trophoblast. Okay, this trophoblast helps in the formation of the placenta. Okay, what about the inner cell mass? The inner cell mass are called as embryoblast. These are called as embryoblast. Okay, so the embryoblast helps in the formation or development of the embryo. Okay, hence the name embryonic pole. The part where your embryoblast is attaching, that is called as embryonic pole. This pole is called as embryonic pole. Okay, next the blastocele. Blastocele is filled with the uterine fluid, and it will give rise to which part? It will give rise to umbilical, amniotic cavity and yolk sac. Okay, and remember again, in the trophoblast we have two types. One is polar trophoblast and the another one is mural trophoblast. Trophoblast we have two types. Okay, the first one is polar trophoblast and the another one is mural trophoblast. Okay, which one is polar trophoblast? The outer cell mass which is in contact with the embryoblast part, this part. Okay, these cells are considered as the polar trophoblast. Okay, and the remaining cell layer, this one. Okay, this is called as this layer. This is called as mural trophoblast. Okay, so if they are asking you to write the short notes on the structure of the trophoblast, you should draw this diagram and you have to mention about all these structure and these points. Okay, any doubts? Any doubts here? Everyone understood the topic? Any doubts you can write. No doubts? Sure. Polar trophoblast, mural trophoblast. Okay. The cells that mean the outer cell mass or the cells which are in contact with your embryoblast part. See, this is the embryoblast part, right? So which is in contact with the embryoblast, they are named as polar trophoblast. Okay, the remaining one which are towards the blastocyst structure, this layer, it is named as mural trophoblast. Okay, so the whole process of cleavage and blastulation. Your blastulation also includes the formation of trophoblast and embryoblast and the amniotic cavity or the blastocyst. Okay, all these events happen in 1 week. Okay, so these events are considered as the first week of development. First week of development. So if you get a question, discuss the events that takes place in the first week of development, you need to mention about the cleavage and formation of blastocyst. Okay, stop talking. So if the question is describe the events that takes place in the first week of development, you need to mention about the cleavage starting from the 2 cell stage to the 64 cell stage, followed by the formation of blastocyst. Okay. So within how many hours this will take place? From the zygote to the 2 cell stage, it will take place in 30 hours. Okay. Then to the 4 cell stage, it will take place between the 40 to 50 hours. The 8 cell stage takes place around 72 hours, and the morula will form after the 96 hours. MCQ question. Okay. So by the time your 32 cell stage reaches into your uterine cavity, it will take around 4 to 5 days. After reaching into the uterine cavity, in the next 2 days, that is on the 6th and 7th day, it will undergo the 64 cell division, then followed by the formation of blastocyst. Remember these points, take down the notes now only because it's your MCQ questions. This is all about the cleavage and formation of blastocyst. Any doubts? No doubts? Any doubts in fertilization? Any doubts? Others, take down the definition and duration. Subdivision, it's up to you. If you want to mention, you can mention. Otherwise, directly you can put a heading as cleavage, then followed by formation of blastocyst. Okay? You can start reading. Yes.
It is a process of repeated mitotic division of zygote within
zona pellucida
in rapid succession.
Yes.
Giving rise to increasing number of smaller cells called
blastomeres. So the cleavage is nothing but the repeated mitotic division to form the number of smaller cells called blastomeres. Okay, next.
The cleavage division lasts for 6 days up to 7 days after fertilization.
Yes. So this will be the first week of development. Next.
Subdivision of cleavage. There are 3 subdivisions in cleavage. They are
Stage of compaction.
Morula,
Blasto. Yes. Next. 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 You also have throat infection. Give it to him.
This results in a rapid increase in the number of cells. These cells are called blastomeres. The division of zygote starts just after fertilization and continues with the zygote passes along the uterine road. During cleavage, the zygote is surrounded by a rather thick zona pellucida.
Take down the point. During the cleavage, during the whole process of cleavage, the zygote is surrounded by the zona pellucida layer. Okay. Next.
The zygote divides to form two cells, of which one is smaller than the other, two cell stage of embryo. The larger cell divides first, giving rise to three cell stage. The smaller cell divides next, and embryo consists of four cells that divide to form eight cells.
Next.
Eight cells further divide to form 16 cells. A cell stage embryo resembles a mulberry fruit and its is termed Morula. In Morula, the blastomeres are very small and
Others, stop talking.
contains very little cytoplasm to survive.
So your morula consists of very small cells. Those are called as blastomeres, and it contain very little cytoplasm. Okay.
The blastomere are enclosed by the zona pellucida. The cells now get arranged into two groups. A group of cells in the center of morula is called inner cell mass, Embryoblast, and those that are present at the periphery of morula are called outer cell mass, Trophoblast.
Okay. Can you see this diagram? Do you see any difference from what I have explained? Can you spot any difference? So in general, when we are studying about the cleavage or trophoblast structures or process, they are going to explain it like this, the way I have explained, okay. But in few textbook, they have also mentioned about 3 cell stage and 5 cell stage, okay. The process for 3 cell stage and 5 cell stage division is, after the fertilization, the zygote will undergo 2 unequal division, okay. Here I have almost shown the equal cell division, right, and I have mentioned it as 2 cell stage. Exactly this 2 cell stage, but it will undergo in a unequal division. One cell will be larger and the another one will be smaller. The one cell which is larger will first undergo one more mitotic division, and the smaller one will remain like this, okay. So this is the larger cell and this is the smaller cell. So next, in the next mitotic division, the larger cell will undergo the division first and it will give us 2 cells, whereas the smaller cell will remain the same. So this stage is called as 3 cell division, okay. After this, in few textbook again they have explained, the small cell is now considered as the large cell, right. So now this will again undergo the one more mitotic division, unequal division sometimes and sometimes equal division, like that they have mentioned. In few textbook they have also mentioned about 5 cell stage also, but right now try to remember only about the 3 cell stage, okay. After that, the 4 cell stage, then followed by 8 and 16, then the process is similar like what I have explained, okay. Next. This is the different cell stages in different animals actually, which is pretty similar to the human cell division only, okay. This is the detail of time duration of your cleavage process. You can take down the notes of this, or it is similar to what I have explained, but their time, like they have given 2 times. There I have mentioned directly 30, 40, 50, 72 followed by 96, right. So here they have given the time duration between 32 to 42, 56 to 72, like that. So any doubts in cleavage? Any doubts? No doubts? Next, blastocyst formation. Okay. So once the morula enters into the uterine cavity, it will be interrupted by the entry of your uterine fluid, which is also known as endometrial fluid, okay. Once the endometrial fluid enters, there will be disruption in the arrangement of the cells. Okay, hence it will give us the outer cell mass and inner cell mass layers, and that kind of formation of cell is called as blastocyst. The endometrial fluid, it is also known as uterine milk. Next,
Trophoblast at one pole, the embryonic pole. The resultant whole structure is now called blastocytes. The embryo gives rise to the embryo, and the trophoblast provide nutrition to the embryo. The blastocyte enlarge in size, and the zona pellicula disappeared. Now the blastocyte become ready to implant.
opposite. So as the cavity enlarges, the outer cell mass layer, that is the trophoblast cells, will get flattened in structure. Okay. Then the point where the inner cell mass is attached to your outer cell mass, that end is called as the embryonic pole. Now the old structure we call it as the blastocyst. Before the implantation, the zona pellucida will be disappeared. Okay. So this is the diagram for the blastocyst formation. You can observe the structures. The opposite to the embryonic pole, they name it as abembryonic pole. Okay. Any doubts? Any doubts in this figure?
एक्सप्लेन कर सकते हैं
Which one?
लास्ट में ब्लास्टोसिस्ट
Hmm.
क्योंकि यहाँ से वो क्लियर दिख नहीं रहा
Okay. Shall I clean this? Okay. The last one. This is your blastocyst structure. Okay. From the 16 or from the 64 cell stage, once the accumulation of your embryonic fluid takes place, the cells will divide. Okay. Now the cells will arrange in the outer layer like this. Okay. This outer layer is called the outer cell mass. Okay. And at any one pole, there will be arrangement of few more cells like this. Okay. Now this is called as inner cell mass. Okay. And this entire cavity, this is called as the blastocele or blastocyst cavity. Okay. So now this pole where your inner cell mass is attached, this pole is called as the embryonic pole. Embryonic pole. Okay. The opposite to this embryonic pole, that means this pole, they call it as abembryonic pole. This is called as abembryonic pole. Okay. This inner cell mass are also called as trophoblast. These are called as trophoblast. Okay. This will give rise to or helps in the formation of placenta, which provides nutrition or required nutrients to the developing embryo. Okay. The inner cell mass is called as embryoblast. This is called as embryoblast. Okay. This embryoblast will helps in the development of the embryo. That means development of the baby. Okay. Later the blastocele, this entire cavity will develop into amniotic cavity and yolk sac. What are these structures? We are going to discuss in the future classes. Okay. Now, the cells which are in contact with your inner cell mass, the layer of cells, the layer of outer cell which are in contact with inner cell, this layer. Okay, this is called as polar trophoblast. Okay, the cells or the layer of outer cells which are not in contact with the inner cell, that means this layer, this is called as mural trophoblast. The same structure. Okay, is it clear now? Any doubts for others? No doubts? Sure? Everyone? No doubts? Okay.